So, firstly, welcome everyone who has joined and thank you for joining. And thank you, Professor David, for joining us. It's a great pleasure for us. So, before beginning with the webinar, I'll tell you a little about the Biology Society. So, the Biology Society is basically like we have tried to create a platform for like minded people who are interested in the fields of biology, various fields. And here we, try to share our knowledge. And as we say, the common quote is with sharing your knowledge increases. So, that is what exactly we are here for. And we do various events, webinars, and we had a conference recently on National Science Day. So that is about us. And to know more, I'll share the website link of our society, and I'll hand over the space to our moderator, Swanzil.
So, welcome everyone. My name is Swanzil Aurora, and I will be the moderator for today's webinar. We are delighted to welcome Professor David Baum today. Professor David is currently working as a professor of botany at the University of Wisconsin-Madison. He completed his PhD from Washington University. His research interests are plant phylogenetics, conceptual issues in evolution, and systematics and the origin of life. His research goal is to understand how evolution works to generate the diversity of living organisms today. In addition to theoretical and conceptual interests, he has focused on evolutionary, developmental, and molecular phylogenetic research on flowering plants. He is the co-author to Tree Thinking: An Introduction to Phylogenetic Biology and has received awards like the Guggenheim Fellowship for Natural Sciences. Professor Baum here with us today to deliver a fascinating webinar on the origin of eukaryotes from the inside out. Also, we will be having a question and answer space at the end, and any doubts during the webinar can be asked on the chat box, which will be taken towards the end. Welcome, Professor David, and I would like to hand over the space to you.
Great. Thank you very much for inviting me. It's fun to be talking to your audience, and that was a very nice introduction. So I am happy to keep my chat window open so I can see if you do have burning questions while I'm talking. Could you just remind me how long you want me I have the floor for? I have forgotten.
So we can have it for 45 minutes to 1 hour, and then
Okay.
it's on you how comfortable you are.
Okay, great. Thank you. I thought that was the case, but I wanted to check. So you are very welcome to put questions or comments. Okay, so what I want to talk about is the origin of eukaryotes, and I'm going to present to you an alternative, a new way of thinking about this that I developed and published maybe 6 years or so ago. So the origin of eukaryotes is arguably one of the most important transitions that has happened in the origin and evolution of life. So how has this transition happened? And I consider it a remarkable transition, but in both senses of the word remarkable. It's remarkable because it happened once, and there really has been no analogous transition that has occurred during the history of life on Earth. The second reason it's remarkable is that it was very impactful, because the eukaryotes, with their more complicated cell structure and all the additional ways in which they can regulate gene expression, have been extremely successful organisms and are the main multicellular organisms on the planet, including us, of course. So it was an important transition, and it's worth trying to understand what happened. So this little sort of schematic tree that I have before you just shows the overall relationships. And we now know that the eukaryote, most of the genome of the eukaryotes is related to these lineages in blue, which are these archaea prokaryotes, and distantly related from the bacteria, the other main branch of the tree of life. So what we're trying to understand today is, how did the eukaryotic cell evolve from a prokaryotic ancestor? So let me first sort of review the differences between eukaryotes and prokaryotes. So eukaryotes, first of all, are much larger than typical prokaryotic cells. So prokaryotic cells have a typical size range of 0.1 to 5 microns, whereas typical eukaryotes have a size range of, of 10 to 100 microns. So there's a size difference. Secondly, there are many novel structures that you find inside a eukaryotic cell. So there's a nuclear compartment with a nuclear envelope, which has pores in it, which are specialized structures. There's the endoplasmic reticulum, there's the Golgi apparatus, there's the vacuole, lysosomes. There's a whole set of membrane-bound compartments that we call collectively the endomembrane system. And then there are structures like flagella, which are complicated structures involved in motility. So they're bigger, they have novel structures. They also have novel processes. So they are, thanks to having a dynamic cytoskeleton and a lot of protein machines that can manipulate and move membranes around, such that they can adjust their shape very dynamically. Eukaryotes are very active cells. So they do lots of things. They can engulf other cells through phagocytosis. Like this is a macrophage chasing a bacterium around in some blood, among some red blood cells. They can exocytose, like the cell on the right, setting out little vesicles. They have all kinds of internal systems that are quite complicated. So, the way that one of the things this shows is that through these novel processes, once eukaryotes evolved, they became the sort of the top predators. They are the lions and tigers of the microbial world. But they also acquired all these other capabilities that allowed them to become large and multicellular. So one of the features that is special about eukaryotes is that eukaryotes have some endosymbions. Now, all the eukaryotes have mitochondria or have evidence of having had mitochondria. There are a few obscure protistan eukaryotes that no longer have mitochondria, but we know that they did have mitochondria because we find mitochondrial genes remaining in the genome. So mitochondria, that while some eukaryotes also have plastids, that's something that occurred later, gave rise to the photosynthetic organisms. I'm not going to talk about those, even though I'm a botanist and I like them. But the mitochondria are very important because the mitochondria were present in the earliest eukaryotes, and which tells us that they originated at or around the time the eukaryotes emerged. And so so the first thing I want to clarify is, where did the mitochondria come from? Because this is one thing that everybody agrees on. And I'm pretty sure you know this already, that it's pretty clear that mitochondria are derived from free living bacteria, specifically a group of bacteria called the alpha proteobacteria. So this was first inferred because they have a lot of biochemical and structural similarities to bacteria. They have similar genome, they have similar ribosomes, and they have similar sensitivities to certain antibiotics. Another very important clue is that within modern cells, it's not been ever observed that mitochondria arise from de novo, from scratch. They always emerge by the division of a prior pre-existing mitochondria. So they function like cells dividing within other cells. But the real strong evidence, the sort of overwhelming evidence, comes from phylogenetic analysis of genes that sit in the mitochondrial genome. And this has been known for a long time. This is an old example, but there are many other studies showing this, that with this particular gene, what you can see is that we have in this bottom clade the mitochondrial genes of a lot of taken from a lot of different eukaryotes. Just as a note, this bottom one, Trichomonas, is one that doesn't have a mitochondrion, but it does have a gene that is related to the mitochondrial genes found in other eukaryotes. And this clade is sister to the alpha proteobacteria on this small tree, and distantly related to the the equivalent, the most closely related gene in the nucleus. So that confirms that indeed that the mitochondria component of a eukaryote is a bacterium that was taken up somehow and became integrated into the eukaryote. So the eukaryotes are really a chimera, a merger of two distinct lineages, the bacterial lineage and a lineage of basically archaeon. So, this is established beyond a reasonable doubt based on the phylogenetic trees. And we also understand that the bacteria coming in and becoming mitochondria confer huge benefits on eukaryotes. The ability to have all these internal membranes that can do electron transport, the ability to take, unlike in nature where bacteria sort of compete for protons to make ATP, here they all have their own little space. They can put their protons, they can turn them into energy, they can turn them into ATP. They also, importantly, the mitochondria, I'll talk again a little bit later, when the introduction of mitochondria provided the key lipids that allow eukaryotes to adopt some of their structural novelties. And they also brought in a number of other important genes, genes for manipulating membranes, like if you've ever heard of dynamin, that gene family came from the mitochondrial lineage. But so we know they came from bacteria, we know they are bacteria. We know they're important, but how did they get inside? So, and that relates to the question of, well, how did all the other membranes get inside? So if you look at a eukaryotic scale in some sort of schematic manner, what you find is that there are many, many internalized internal membranes. And the question then comes, how did the mitochondria and all of these other membranes end up inside the eukaryotic cell? And that's really what I'm going to focus on today. Now, traditionally, the story is assumed that these membranes were formed from the outside in. So what that means is to say that the precursor archaeal cell became larger and acquired the ability to move membranes. And what it did was it began invaginating, internalizing membranes. For example, when eating prey items by engulfing them. And in so doing, it brought membranes inside the cell. And those membranes structures, the vesicles and tubules that would have come in, ultimately fused together and became organized, including the production of an internal compartment around the chromosomes, around the genetic material to protect it, if you will. That's the hypothesis. And the other components became sort of specialized different cellular functions like secretion, or digestion, or recycling, and so on. So that's been the traditional view. But the view I want to propose to you is one that we published in 2014. So I guess it's more than 6 years ago, which is the inside-out hypothesis. So what the inside-out hypothesis speculates is that actually it's the other way around. That the original cell, the original archaeal cell, it never grew larger. Instead, it generated through holes in its cell wall extracellular protrusions called blebs, the convenient term. And these blebs ultimately became more and more complicated over time and fused together and became organized to generate what was formerly an extracellular compartment, the cytoplasm, and a bounding cell membrane. So all of the key events in this involve folding membranes in an outward direction, whereas in the outside-in model, the early steps involve folding membranes in an inward direction. And now these are very different and involve the folding of membranes in these two directions in living life uses completely different sets of proteins because of the way in which you're bending them. And so it seems we've argued it was worth considering that in fact this is the direction in which things happened, not the direction that everybody had previously been assuming, which is from the outside in. And so I will say at this point that I really do welcome questions or clarifications. If I'm going too fast, I'm going too slow, you're welcome to type something or interrupt me. Okay, so part of when we published this paper, one of the things that we highlighted as a sort of starting point for this was the observation that in the archaeons, so these are the believed to be the source of the cell, all of the cell except for the mitochondria, they usually, almost always, produce extracellular protrusions of some kind or another. Now, the functions of these structures are not known in most cases, but they are almost always produced. So here are a couple of examples. This is a transmission electron microscope showing these little sort of peg-like protrusions. And this is a scanning electron microscope of Thermococcus showing these things called nanospheres, which are little membrane bubbles that stick out of the cell. Now, typically, in these extracellular protrusions are pinched off and released as exosomes, but nonetheless, we noticed that there are very many of these instances, actually, particularly in the group of archaea that are most closely related to the eukaryotes. And there are no known cases of internalization of membranes in the archaeans. And so that seemed like suggested that we should be thinking positively about a model that focuses on outward projections first, rather than putting that as a sort of putting the inward ones as a starting point. So with the hypothesis that I developed was that the ecological function of these extracellular protrusions was to interact with free-living bacteria, mutualistic bacteria that the host cell could be exchanging materials with. And there's some basis for this in modern life, in that there are quite a number of associations between archaeal cells and bacterial cells that appear to be interdependent and mutualistic. So this is an example of a very large archaeal, filamentous archaeal species called Giganthauma, that has these sort of these are actually two archaeal cells sort of stuck together. And they are usually found with a coating of a gamma proteobacteria. So this leads to the idea that if you have extracellular mutualists, there might be some benefit to expanding the surface area of the cell membrane in order to promote exchange of materials. So let me now talk you through the hypothesis. So the idea is we start off with an archaeal cell with a cell wall, and that has pores in the cell wall through which it protrudes little peg-like protrusions. And these help exchange materials with extracellular bacteria shown in the red dots. So the idea is that selection could now act to prevent the fission, the separation of these extracellular protrusions as exosomes, so they get stabilized and they start getting bigger, because they're like the roots of the tree or the mouth of the animal. They are the way in which material are being exchanged with the extracellular symbiont. So they would get bigger. And this would do two things. One thing is that it would increase the area of association, but it would also serve to eventually trap the bacteria close to the host cell, meaning that those bacteria would be protected from the outside world and would also be, they would not be lost as readily. And in fact, when the cell divides, they could be passed on. So the way to think about this, or a way to think about this or visualize this, is to think about it topologically as, you know, the cell body is like the Mars lander. And those airbags that Pathfinder used to land on Mars, those are like balloons. Now that person could stick their hand between those balloons, and they could be trapped between those balloons. They get on around the outside of the lander. They could be little, I don't know, you could put some soccer balls in there or something, or cricket balls. Yeah, so that's topologically what's happening. So the idea then is that these extracellular protrusions would sort of fold around and stick down to the outside of the former cell body, tightening up those connections, and they would still at this point maybe be the remainder of a cell wall. Of course, translation, the conversion of RNA and RNA message into protein through ribosomes would be happening both in the nucleus and in the blebs. But over time, this would become resolved to give rise to the modern formation by losing that original cell wall, by the formation of a continuous cell membrane on the outside. And we have some ideas how that could have happened. By preventing translation in the nucleus. So now, as you probably know, in a modern eukaryote, the ribosomes are not fully mature and able to do translation until they're in the cytoplasm, although there is some evidence of occasional translation in the nucleus. But basically, translation has been suppressed in the nucleus, which allows for additional layers of gene regulation and probably contributes to the regulatory complexity of eukaryotes. And then at some point, the mitochondria, which were formerly trapped in what is basically the endoplasmic reticulum. Let me go back a second. So you can they start off in the gaps between these blebs, which corresponds to the future endoplasmic reticulum. But at some point, they enter the cytoplasm, so that they're no longer in the ER, they're now outside the ER. Now there's a couple of ideas that we have for how that could have happened. Remembering that the bacterium that was engulfed was a proteobacterium, and proteobacteria have two membranes. The outer membrane is a funny membrane. It's sort of a porous membrane, but they do have two membranes. So there are a couple of ways, 3 ways we can think of that you could end up with mitochondria being fully inside the cytoplasm. The first possibility is that the ER is punctured and they sort of get in. The ER that they were in is no longer there. Second possibility is that they actually are still effectively in a little outer layer of ER, and one of the membranes, the outer membrane, presumably was lost. And the third possibility is some kind of fusion where the outer membrane of bacteria and the membrane that was the ER membrane, the host membrane, sort of became one in some way. So that's the basic structural hypothesis, and I'm going to and we view this as being kind of a logical set of evolutionary drivers. So at the very beginning, we have selection for improving the surface area of exchange between the host and the mitochondria, the future mitochondria, the protomitochondria. Then by expanding these blebs, we also now have selection for protecting these essential resources. You know, in a sense, you could think of the host cell as farming these mitochondria, and you want to keep your crops safe and you want to keep them for yourself. And make sure that when you divide, both your daughter cells will get a subset of your crop, your seeds. And then the proliferation of the cytoplasmic compartment and the development of a full nuclear pore. I don't know where that word came from, of a full cell membrane would, we argued, be driven by selection for protection of the secretory machinery. So that's the hypothesis, and I'm going to spend the rest of the time talking through some of the evidence that supports the inside-out model. But I actually, if it's okay with the organizers, I would suggest that it wouldn't be bad to spend a couple of minutes answering any questions that might have come up already. Would that be okay?
Yeah, sure. So if anybody has any questions, they can ask.
You can ask verbally, or you can, if you don't feel comfortable talking, you're welcome to write on the chat.
So in the inside-out theory, you mentioned that there's this formation of blebs. So, like, what is the thing that stimulates its formation?
Well, I guess I'm not sure the word stimulate could have two meanings. You could mean in an evolutionary sense, like why was this structure developed? And the argument there is that these cells are already making extracellular protrusions. And so they would, what the key change was stopping them being pinched off, ensuring that they could remain stably outside the cell. And the evolutionary idea is that the reason for that was that this was beneficial because it facilitated exchange of materials with the protomitochondria, the extracellular bacteria. Is that clear?
Yes, got it, sir. Thank you.
Yeah, thanks for the question. Okay, if there's no questions, I will keep going. Okay, so there's how could we assess this hypothesis and compare it to how could we compare the inside out and the outside in alternatives? So I'm going to talk about three kind of approaches we take. The first is sort of looking at the features of mitochondria and those prior to endosymbiosis. Then we can look at some of the features of living eukaryotes to see if they just sort of seem to align with one model or the other. And then the last thing I'll mention is we can look for diversity, new organisms that may help bridge the gap. So the key feature of bacteria that I think we need to talk about are the membranes. So bacterial membranes are very similar to eukaryotic membranes. And given that most of you are biotechnology students, I imagine that you're aware that that they are composed of ester-linked phospholipids. They have long straight chain fatty acids, and the backbone is a glycerol 3-phosphate. Now, you perhaps know that archaea have very different membranes. They are isoprenoids, are the sort of the hydrophobic groups. They're ether-linked, not ester-linked, and the backbone is a glycerol 1-phosphate. So the question then is, given the phylogenetic tree, how do we explain the fact that archaea, which if you know the term are a paraphyletic group, how could they have one situation while bacteria and eukarya have the other condition? And the answer is pretty clearly that the membrane chemistry of eukaryotes was provided to them by the mitochondria. And so this little complicated chart here just shows which bacterial groups or other groups, archaeal groups, have the closest relatives of key classes of genes in eukaryotes. And so, if you look at lipid transport and metabolism, the largest fraction of those genes came from alpha proteobacteria. So this leads us to the conclusion that what actually happened was that during the origin of eukaryotes, this is a more recent phylogenetic tree, I've added a few branches, some of which I'll talk about later, that on this eukaryotic lineage, that's when the mitochondria arose, and that's when the lipids took over. So this lipid revolution was a very important transition. And I would suggest to you argues against the outside-in model. So for the outside-in model, how would we have to explain this? So we would have to say the following. We'd have to say that the host cell, the archaeal cell, somehow engulfed a mitochondrion. Now, when it engulfed the mitochondrion, it would be using, it would already therefore have to have highly dynamic membranes in order to be able to envelop and take in this bacterial that's going to become the mitochondrion. So what this would imply is that all the membrane manipulating machinery of the host cell evolves before the interaction with the mitochondria. And then the mitochondria come in, and there's a whole lipid revolution. And now all of those membrane manipulating proteins would have to sort of be retrofitted. They would have to learn how to work with these new membranes, which just seems very unlikely. So in contrast, the inside-out model, this makes complete sense, because what happens here is that the association occurs prior to the evolution of any of these membrane complexities. So you have this close affiliation of the mitochondria with the host cell, allowing plenty of opportunities for the exchange of genes and actually physical lipids through contacts that could result in the lipid revolution. And only later does you end up having to, do you add and embellish upon your repertoire of membrane manipulating proteins using the new membrane types. And so I think that this order of events is much more plausible than the outside-in version, which requires that you first learn how to move membranes around, and then you have to learn it again when the membranes change. There's also reasons to think that the actual physical chemistry of archaeal lipids are not as conducive to this, to being able to do things like phagocytosis as the phospholipid membranes of bacteria. Okay, so that was just sort of an argument based on sort of the characteristics brought in by mitochondria. There's also a number of structures under the headline I would call consilients. So just features of eukaryotes that, once you are thinking in this inside-out way, seem sort of to fit. So an obvious simple one is that if you look at a nucleus, it doesn't have internal membranes. It looks like a prokaryote sitting inside a mass of membranes. And so that is obviously inevitable given the inside-out model. It's not obvious under the outside-in model why the nucleus shouldn't have, at least in some species, internal membranes. And incidentally, this is really what was the first reason that I proposed this idea. I'll get into a couple more specifics, but I'm not going to, because I don't want to spend too long on the details. There are some other features of eukaryotic cell biology that align and can be sort of predicted from the inside-out model that are less obviously predictable from the outside-in model. So one example is the nuclear pore complex. So under our model, the nuclear pore complex is descended from membranes that would have stabilized these protrusions and prevented these protrusions from being pinched off. And so they would have been controlling positive membrane curvature on the inside of the cell at the base of each protrusion. Now, when that protrusion has to form a bleb, and the bleb sticks down on the outside of the cell envelope, the cell wall, the S layer, we would have exactly mirror image positive curvature on the outside now. And this hypothesis predicts that nuclear pores should be mirror image structures on the inside and outside. And in fact, they are. Now, it could happen under the outside-in model, but it's not predicted by the outside-in model that they would have this particular symmetry. Similarly, perhaps you can see that the prediction would be that the control of the separation of the two sides of the nuclear envelope would likely involve proteins coming from the two sides that interact with something in the middle. It used to be the cell wall, the S layer, but once the cell wall goes, maybe we would have some residue of that, some protein linkers that link these two pieces to one another. And in fact, we do. And they also glycosylated in the way that archaeal cell walls are glycosylated. So they're called link complexes that control the spacing of the two membranes. And as I said, they have features that are predictable under the inside-out model and sort of not obviously predictable under the outside-in. And I mentioned briefly, I don't want to, a couple of other things. One thing that the inside-out model predicts is that in a eukaryotic cell, the entire endoplasmic reticulum corresponds to the formerly exterior of the cell. And if you go back to that Mars rover, you can tell that that whole ER, however complicated it is, all connected together. It's all one system. Whereas the cytoplasm is kind of discontinuous. And one of the things that cell biologists were aware of is just how amazingly continuous ER usually is. That, even in gigantic cells, like muscle cells, the ER functions like one great big system, connected system. And they've also noticed that cytoplasm can be surprisingly resistant to diffusion. That you can have domains within it. What looks like one cytoplasm can be quite different, and that's why like development of phenocytic Drosophila embryos is just looks like one big bag of cytoplasm, but there are very definite gradients of gene expression that can be seen in that space. And then we know that mitochondria often have intimate associations with ER, which you would predict under this model. So, and then the last fact it really was not something that I would have predicted when we published this paper initially, that we would discover actually new organisms that are more closely related to eukaryotes that can shed light on this transition. And so in 2015, there was the first evidence from genome sequence data of the existence of a new group of archaea called the Asgard Archaea. They were only known from genomic data until last year, when they finally, a group in Japan managed to culture them, but they couldn't culture them alone. They had to co-culture them with extracellular symbionts, a different species that they seem to be entirely dependent upon. And so, so this merger here is sort of we can get a bit more of insight into it by looking at these living Asgard Archaea that are more closely related to eukaryotes than they are to other archaea. And when we look at these, we find that the cells make extracellular protrusions, and they make these long extracellular structures that seem to facilitate interacting with extracellular. These are actually a different species of archaeon that they depend on, and they cannot be cultured without these other archaea. So to my eye, this is entirely consistent with our model, suggesting that the ancestor was already using the external space for interactions with other microbes. One particular lineage was associating with bacteria rather than archaea, and that external space ultimately proliferated to the point of becoming the cytoplasm. So I would summarize by saying that the inside-out theory is at least as plausible as the outside-in model. And I would contend that even if it is proven false, I think it will stimulate, and already has stimulated, new lines of empirical research. And I will tell you just personally that when I started writing this paper, which was probably 2013, I really was not at all convinced that the theory was correct. I thought it just needed to be presented as an alternative. When we published it, I was asked by various people, do I really think it's true? And I said 50-50. But over time, and particularly with the discovery of these new Asgard Archaea and the characterizations of how they work, I am now really quite convinced that actually the inside out model is correct. But I'll have to let you decide if you agree with that. Now, before I completely end, I did want to give you some context here, because I think you might find this interesting given where you are in your careers. So the paper was published in 2014, but the idea is much, much older than that. So I initially had this idea when I was an undergraduate student, and I was asked to write an essay on the evolution of eukaryotes, and I just said, well, here's an alternative. I didn't publish it. I didn't believe the data were strong enough. My advisors were saying, yeah, this is just speculation. It doesn't have any, there's no reason to believe this. I actually worked for a time in the lab of Lynn Margulis, who some of you may have heard of, who was famous for her work in developing the endosymbiotic theory. And she was like, it doesn't really have legs yet. But then in 2014, when I was trying to establish my credentials in the Origin of Life field, which I also work in, I decided to work it up, and I began collaborating with a cell biologist I know quite well. He's actually my cousin. His name's Buzz. He's based in Cambridge, England. And so the two of us sort of fleshed out the hypothesis. We published this paper, and it caused a surprising amount of attention, certainly more than most of my work, let's just say. So I guess what my last sort of message to you, this is a scan of my original essay, that I guess my last message to you is that you may at times feel like, you are the lowly undergrads and there are people like me or these, professors, but you should remember that ideas come at any stage in one's career, and that sometimes people who are earlier in their careers actually can see things in a different way and not be constrained in their thinking. And so I really think that science needs naive students and outsiders all the time, people with different perspectives, different backgrounds, and not locked in ideas. So with that, I guess I'll just say thank you for inviting me, and happy to take any questions. Great. Looks like some people have been preparing questions. I like that. So more about so there are other first thing to say is that I think we have a bias. We tend to think of organisms as sort of units, each species and each instance, each individual of that species is just sort of a separate thing. But I think we now know that mutualism and symbiosis is everywhere, that all living things sort of interdepend upon other living things. So there are many instances of microbes engaging in endosymbiosis. So, and there's many subsequent cases involving eukaryotes. So there are many eukaryotes that have taken up and associate with bacteria, for example, plastids, chloroplasts, and so on. There are a number of examples of important metabolic endosymbiotic bacteria, like, for example, aphids have them. But then there are also cases of associations between pairs of prokaryotes, but they don't get inside one another because they don't have the flexible membrane. There is one weird case involving an archaeon called a pair of archaea, where one is sort of inside the other. So it was inside the cell wall, but it's not inside the membrane. It's called nanoarchaeum, and it's inside. I've forgotten the name of the host. I'm blanking out on that. Okay, so let me quickly answer Sakshi's question, and then I would love to hear someone speak. So the question was, the question is basically, had there been no bacterial derivation of mitochondria, how could the cell make do? I mean, it's a little hard to imagine how the eukaryote could have evolved without mitochondria, because so much of the work of the cell is driven by large amounts of ATP to manipulate those membranes. And without mitochondria, it's sort of hard to see how they could do it. Now, but you could, if you wanted to come up with a story, you would say that they would create by internalizations little structures that could do electron transport, trap the protons, and could do basically what a mitochondrion does. In which case, I suppose they could make do on that. But mitochondria are great because they can divide. You can, if a cell needs more energy, can do more aerobic respiration, they can make lots of mitochondria quickly. If they need fewer, then they can eat a few. So they're very dynamic in a way that it's harder to be with your own membranes, I think. But who knows? Okay, Ashwin, would you like to ask your question?
Yes, am I audible?
Yes, you are.
So my question is, suppose now we have colonies and cells communicating with one another in various multicellular organisms today. Now, if I extend, how can I possibly extend this concept to the establishment of cell connections and possibly multicellularity, and also how can I bifurcate between plant and animal like organisms so if I go forward with this story to fungi and all those fungi, plants and animals possibly?
Yeah. Well, so I think this has some interesting implications for how we think about multicellular organisms. So I think that we've tended to view the cell bounded by the cell membrane as the unit. And then that cell divides, and then those two daughter cells stick together. And that's how you eventually get a multicellular organism. But in fact, a lot of things that we call multicellular don't actually fully do that. They are more like a space with many mitochondria. So there are we call that a syncytium. Maybe I should write that in here because I haven't got it on a slide. And many of you know with that a syncytium. I guess it's correcting my spelling, which is great. A syncytium is, a cytoplasmic space that contains many nuclei, and these are very, very common in multicellular organisms. I mentioned Drosophila embryos. Many plant embryos also are cenocytic. There are many algae that are cenocytic their whole life. Typical fungal cells often contain at least 2 nuclei. So it's actually almost easier to rethink what it is to be multicellular as not being multicellular, but being multinucleate. Because if you have a cell, I actually I had an image of this and I took it out, which I now regret. But if you take a cenocytic space where each of those nuclei is surrounded by its own cytoplasmic domain, then you can understand how that can function very much similarly to not a cenocytic space, but a multicellular space where there are membranes between each. And I guess the last thing, I think you actually maybe you said this, but in a plant cell, all the cells are actually continuous. Almost all the cells are continuous because of these little tubes that connect every cell called plasmodesmata. So when plant cells divide, they don't fully divide. They leave little channels between those cells. So the cytoplasm is still continuous. To some degree, there can be ER tubules that connect them, but they still can remain in different genetic states because sharing, not having a cell membrane separating two things doesn't mean that they're now completely free to interact, because each nucleus controls its sort of surrounding cytoplasm. Did I kind of answer your question, Ashwin?
Yes, sir, you did. Yes, sir. Thank you so much.
You're very welcome. Tardigrades are animals. I haven't worked on them. They are a group of animals that are closely related to the arthropods, but are not arthropods. And they're famous because among animals, they're among the most resistant of desiccation. They can dry down almost completely and then be regenerated. Yeah. I guess
Sir,
I
you
should.
mentioned somewhere Asgard Archaea.
Yes.
Sir, is it like real based upon like a real Asgard something or what?
I may have. So you want me to talk about the Asgard Archaea? What did you ask about them? I just couldn't quite hear. Say it again.
Sir, it was actually, I wanted to ask about Asgard Archaea. Like, why is it called by the name of Asgard?
Oh,
Because
okay,
as
yeah,
we know, Asgard, it's in Thor, the
exactly.
movie.
No, exactly. Yeah, you're quite right. So let me go back. I don't put all the slides in, but let me just go back a second to this. So this paper in 2015, what happened was these investigators were working at a particular location in the, I think, the Baltic Sea or the North Sea, at an underwater site that is called locally Loki's Castle. And Loki's Castle, they collected some sediment and they found these new genomes that they called the Loki Archaeota that are sister to the Archaea. Subsequent to that, and I unfortunately didn't bring a tree here to show you, people have discovered many other genomes of other things that are not Loki Archaeota, but are sort of related to them. And each of the groups has now been given a name from Norse mythology. So there's the Odin Archaeota, there's the Heimdall Archaeota, there's the Frey Archaeota. And so this whole clade is called collectively now the Asgard Archaeota. The last study I looked at suggests that the most, so the cultured individual that I showed you a picture of, that is a Loki Archaeota. It turns out Loki Archaeota are no longer the closest living relatives of eukaryotes. More closely related than Loki Archaeota seem to be things like the Heimdall Archaeota, and nobody has yet managed to look at those and culture those. So a lot of groups around the world are frantically trying to isolate and study various members of the Asgard Archaeota. And we've only known about them for 5 years, and they are very difficult to culture because they come from often from marine sediments, and they grow very, very slowly. And like this one, they often need other microbes in order to live, and so you can't get them into pure culture. Yeah, so they are all named from Norse mythology.
All right, thank you, sir.
You're welcome.
So thank you so much, Professor David.
Thank you for inviting me, and I hope you have a good rest of your night, evening. Okay.
Thank you so much.
Goodbye.